Transcription, biology. Transcription is the first of several steps of DNA-based gene expression. Gene is a fragment of DNA that encodes for a protein. Some genes encode for a non-coding mRNA such as microRNA or long non-coding RNA, in which a particular segment of DNA is copied into RNA, especially mRNA, by the enzyme RNA polymerase. Both DNA and RNA are nucleic acids, which use base pairs of nucleotides as a complementary language. During transcription, a DNA sequence is read by an RNA polymerase, which produces a complementary, antiparallel RNA strand called a primary transcript. Transcription proceeds in the following general steps. The stretch of DNA transcribed into an RNA molecule is called a transcription unit and encodes at least one gene. If the gene encodes a protein, the transcription produces messenger RNA, mRNA. The mRNA, in turn, serves as a template for the protein synthesis through translation. Alternatively, the transcribed gene may encode for non-coding RNA such as microRNA, ribosomal RNA, RNA, transfer RNA, tRNA, or enzymatic RNA molecules called ribozymes. Overall, RNA helps synthesize, regulate, and process proteins. It therefore plays a fundamental role in performing functions within a cell. In virology, the term may also be used when referring to mRNA synthesis from an RNA molecule, i.e., RNA replication. For instance, the genome of a negative-sense single-stranded RNA, sRNA, virus may be template for a positive-sense single-stranded RNA, sRNA+. This is because the positive-sense strand contains the information needed to translate the viral proteins for viral replication afterwards. This process is catalyzed by a viral RNA replicase. Background. A DNA transcription unit encoding for a protein may contain both a coding sequence, which will be translated into the protein, and regulatory sequences, which direct and regulate the synthesis of that protein. The regulatory sequence before, upstream from, the coding sequence is called the 5' untranslated region, 5'U.T.R. The sequence after, downstream from, the coding sequence is called the 3' untranslated region, 3'U.T.R. As opposed to DNA replication, transcription results in an RNA complement that includes the nucleotide uracil, U, in all instances where thymine, T, would have occurred in a DNA complement. Only one of the two DNA strands serve as a template for transcription. The antisense strand of DNA is read by RNA polymerase from the 3' to the 5' during transcription, 3'5". The complementary RNA is created in the opposite direction, in the 5' direction, matching the sequence of the sense strand with the exception of switching uracil for thymine. This directionality is because RNA polymerase can only add nucleotides to the 3' of the growing mRNA chain. This use of only the 3'5" DNA strand eliminates the need for the Okazaki fragments that are seen in DNA replication. This also removes the need for an RNA primer to initiate RNA synthesis, as is the case in DNA replication. The non-template, sense, strand of DNA is called the coding strand, because its sequence is the same as the newly created RNA transcript, except for the substitution of uracil for thymine. This is the strand that is used by convention when presenting a DNA sequence. Transcription has some proofreading mechanisms, but they are fewer and less effective than the controls for copying DNA. As a result, transcription has a lower copying fidelity than DNA replication. Major steps. Transcription is divided into initiation, promoter escape, elongation, and termination. Initiation. Transcription begins with the binding of RNA polymerase, together with one or more general transcription factors, to a specific DNA sequence referred to as a promoter to form an RNA polymerase promoter closed complex. In the closed complex, the promoter DNA is still fully double-stranded. RNA polymerase, assisted by one or more general transcription factors, then unwinds approximately 14 base pairs of DNA to form an RNA polymerase promoter open complex. In the open complex, the promoter DNA is partly unwound and single-stranded. The exposed, single-stranded DNA is referred to as the transcription bubble. RNA polymerase, assisted by one or more general transcription factors, then selects a transcription start site in the transcription bubble, binds to an initiating NTP and an extending NTP, or a short RNA primer and an extending NTP, complementary to the transcription start site sequence, and catalyzes bond formation to yield an initial RNA product. In bacteria, RNA polymerase holoenzyme consists of 5 subunits, 2 alpha subunits, 1 beta subunit, 1 beta subunit, and 1 omega subunit. In bacteria, there is one general RNA transcription factor known as a sigma factor. RNA polymerase core enzyme binds to the bacterial general transcription, sigma, factor to form RNA polymerase holoenzyme and then binds to a promoter. RNA polymerase is called a holoenzyme when sigma subunit is attached to the core enzyme, which is consist of 2 alpha subunits, 1 beta subunit, 1 beta subunit only. In archaea and eukaryotes, RNA polymerase contains subunits homologous to each of the 5 RNA polymerase subunits in bacteria and also contains additional subunits. In archaea and eukaryotes, the functions of the bacterial general transcription factor sigma are performed by multiple general transcription factors that work together. In archaea, there are 3 general transcription factors, TBP, TFP, and TFE. In eukaryotes, in RNA polymerase II-dependent transcription, there are 6 general transcription factors, TFIA, TFIB, an ortholog of archaeal TFP, TFID, a multi-subunit factor in which the key subunit, TBP, is an ortholog of archaeal TBP, TFIE, an ortholog of archaeal TFE, TFIV, and TFIE. The TFID is the first component to bind to DNA due to binding of TBP, while TFIE is the last component to be recruited. In archaea and eukaryotes, the RNA polymerase promoter closed complex is usually referred to as the pre-initiation complex. Transcription initiation is regulated by additional proteins, known as activators and repressors, and, in some cases, associated coactivators or co-repressors, which modulate formation and function of the transcription initiation complex. Promoter escape. After the first bond is synthesized, the RNA polymerase must escape the promoter. During this time there is a tendency to release the RNA transcript and produce truncated transcripts. This is called abortive initiation, and is common for both eukaryotes and prokaryotes. Abortive initiation continues to occur until an RNA product of a threshold length of approximately 10 nucleotides is synthesized, at which point promoter escape occurs and a transcription elongation complex is formed. Mechanistically, promoter escape occurs through DNA scrunching, providing the energy needed to break interactions between RNA polymerase holoenzyme and the promoter. In bacteria, it was historically thought that the sigma factor is definitely released after promoter clearance occurs. This theory had been known as the obligate release model, however later data showed that upon and following promoter clearance, the sigma factor is released according to a stochastic model known as the stochastic release model. In eukaryotes, at an RNA polymerase II-dependent promoter, upon promoter clearance, TFIE phosphorylates serine 5 on the carboxy terminal domain of RNA polymerase II, leading to the recruitment of capping enzyme, CE. The exact mechanism of how CE induces promoter clearance in eukaryotes is not yet known. Elongation. One strand of the DNA, the template strand, or non-coding strand, is used as a template for RNA synthesis. As transcription proceeds, RNA polymerase traverses the template strand and uses base pairing complementarity with the DNA template to create an RNA copy, which elongates during the traversal. Although RNA polymerase traverses the template strand from 3-5, the coding, non-template, strand and newly formed RNA can also be used as reference points, so transcription can be described as occurring 5-3. This produces an RNA molecule from 5-3, an exact copy of the coding strand, except that thymines are replaced with uracils, and the nucleotides are composed of a ribose, 5-carbon, sugar where DNA has deoxyribose, 1 fewer oxygen atom, in its sugar phosphate backbone. mRNA transcription can involve multiple RNA polymerases on a single DNA template in multiple rounds of transcription, amplification of particular mRNA, so many mRNA molecules can be rapidly produced from a single copy of a gene. The characteristic elongation rates in prokaryotes and eukaryotes are about 10 to 100 NTS per second in eukaryotes. However, nucleosomes act as major barriers to transcribing polymerases during transcription elongation. In these organisms, the pausing induced by nucleosomes can be regulated by transcription elongation factors such as TFIEs. Elongation also involves a proofreading mechanism that can replace incorrectly incorporated bases. In eukaryotes, this may correspond with short pauses during transcription that allow appropriate RNA editing factors to bind. These pauses may be intrinsic to the RNA polymerase or due to chromatin structure. Termination. Bacteria use two different strategies for transcription termination: row-independent termination and row-dependent termination. In row-independent transcription termination, RNA transcription stops when the newly synthesized RNA molecule forms a GC-rich hairpin loop followed by a run of US. When the hairpin forms, the mechanical stress breaks the weak rooted bonds, now filling the DNA R.N.A hybrid. This pulls the poly-U transcript out of the active site of the RNA polymerase, terminating transcription. In the row-dependent type of termination, a protein factor called Rho destabilizes the interaction between the template and the mRNA, thus releasing the newly synthesized mRNA from the elongation complex. Transcription termination in eukaryotes is less well understood than in bacteria, but involves cleavage of the new transcript followed by template-independent addition of adenines at its new 3' end, in a process called polyadenylation. Inhibitors. Transcription inhibitors can be used as antibiotics against, for example, pathogenic bacteria, antibacterials, and fungi, antifungals. An example of such an antibacterial is rifampicin, which inhibits bacterial transcription of DNA into mRNA by inhibiting DNA-dependent RNA polymerase by binding its beta subunit, while 8-hydroxyquinoline is an antifungal transcription inhibitor. The effects of histone methylation may also work to inhibit the action of transcription. Endogenous inhibitors in vertebrates. The majority of gene promoters contain a CpG island with numerous CpG sites. When many of a gene's promoter CpG sites are methylated, the gene becomes inhibited, silenced. Colorectal cancers typically have 3 to 6 driver mutations and 33 to 66 hitchhiker or passenger mutations. However, transcriptional inhibition, silencing, may be of more importance than mutation in causing progression to cancer. For example, in colorectal cancers about 600 to 800 genes are transcriptionally inhibited by CpG island methylation. See regulation of transcription in cancer. Transcriptional repression in cancer can also occur by other epigenetic mechanisms, such as altered expression of microRNAs. In breast cancer, transcriptional repression of BRCA1 may occur more frequently by overexpressed microRNA 182 than by hypermethylation of the BRCA1 promoter. See low expression of BRCA1 in breast and ovarian cancers. Transcription factors. Active transcription units are clustered in the nucleus, in discrete sites called transcription factories or euchromatin. Such sites can be visualized by allowing engaged polymerases to extend their transcripts in tagged precursors, BRUTP or BRU, and immunolabeling the tagged nascent RNA. Transcription factories can also be localized using fluorescence in situ hybridization or marked by antibodies directed against polymerases. There are 10,000 factories in the nucleoplasm of a HeLa cell, among which are 8,000 polymerase II factories and 2,000 polymerase III factories. Each polymerase II factory contains 8 polymerases. As most active transcription units are associated with only one polymerase, each factory usually contains 8 different transcription units. These units might be associated through promoters and/or enhancers, with loops forming a cloud around the factor. History. A molecule that allows the genetic material to be realized as a protein was first hypothesized by François Jacob and Jacques Monod. Severo Ochoa won a Nobel Prize in Physiology or Medicine in 1959 for developing a process for synthesizing RNA in vitro with polynucleotide phosphorylase, which was useful for cracking the genetic code. RNA synthesis by RNA polymerase was established in vitro by several laboratories by 1965, however, the RNA synthesized by these enzymes had properties that suggested the existence of an additional factor needed to terminate transcription correctly. In 1972, Walter Fiers became the first person to actually prove the existence of the terminating enzyme. Roger D. Kornberg won the 2006 Nobel Prize in Chemistry for his studies of the molecular basis of eukaryotic transcription. Measuring and detecting. Transcription can be measured and detected in a variety of ways. Reverse transcription. Some viruses, such as HIV, the cause of AIDS, have the ability to transcribe RNA into DNA. HIV has an RNA genome that is reverse transcribed into DNA. The resulting DNA can be merged with the DNA genome of the host cell. The main enzyme responsible for synthesis of DNA from an RNA template is called reverse transcriptase. In the case of HIV, Reverse transcriptase is responsible for synthesizing a complementary DNA strand, cDNA, to the viral RNA genome. The enzyme ribonuclease H then digests the RNA strand, and reverse transcriptase synthesizes a complementary strand of DNA to form a double helix DNA structure, cDNA. The cDNA is integrated into the host cell's genome by the enzyme integrase, which causes the host cell to generate viral proteins that reassemble into new viral particles. In HIV, subsequent to this, the host cell undergoes programmed cell death, or apoptosis of T cells. However, in other retroviruses, the host cell remains intact as the virus buds out of the cell. Some eukaryotic cells contain an enzyme with reverse transcription activity called telomerase. Telomerase is a reverse transcriptase that lengthens the ends of linear chromosomes. Telomerase carries an RNA template from which it synthesizes a repeating sequence of DNA, or junk DNA. This repeated sequence of DNA is called a telomere and can be thought of as a cap for a chromosome. It is important because every time a linear chromosome is duplicated, it is shortened. With this junk DNA or cap at the ends of chromosomes, the shortening eliminates some of the non-essential, repeated sequence rather than the protein encoding DNA sequence that is farther away from the chromosome end. Telomerase is often activated in cancer cells to enable cancer cells to duplicate their genomes indefinitely without losing important protein-coding DNA sequence. Activation of telomerase could be part of the process that allows cancer cells to become immortal. The immortalizing factor of cancer via telomere lengthening due to telomerase has been proven to occur in 90% of all carcinogenic tumors in vivo, with the remaining 10% using an alternative telomere maintenance route called ALT, or alternative lengthening of telomeres route called ALT, or alternative lengthening of telomeres route called ALT, or alternative length